Welcome. My name is Richard Bacchius. I'm glad to be able to start the program off. We're here for the annual Herbert Newby McCoy Award Lecture. This is always a great day, and I think we're really going to enjoy this lecture. And if you've got your program, you'll notice in the back we have all of the previous McCoy Award awardees and one sentence description of them, and I know a number of them were able to make it today, so if the previous award winners would please stand, let's give them a round of applause. Come on, Nick. Thank you. Last things first, so I want to make sure that after we have the Q&A, we're going to have a reception out the door and to the left in the galleries, the Stewart Center Gallery, so please join us all there when we're done with the program. So if you'll note in the front of the program, there's a description of the award, and so I thought I'd just kind of highlight the key points here. It was established in 1964 by Mrs. McCoy in honor of her husband, a distinguished Purdue alumnus. Mrs. McCoy created this annual award to celebrate her husband's lifelong interest in Purdue and to recognize researchers in the natural sciences who have made the greatest contribution in the year. Nominations for the McCoy Award are submitted by peers and are selected by a committee appointed by the president. Before we introduce Connie Weaver, I'd like to introduce Howie Zelaznik, currently our Associate VPR. Howie's been at Purdue since 1979. He's a professor in health and kinesiology with an extensive publication record in the area of human motor control. He's also been the Associate Dean for two colleges, College of Liberal Arts, as well as the new College in Health and Human Services. He's going to introduce our awardee this year because he participated in the nomination of Connie for this recognition. Howie.
Thank you, Richard. Before I introduce Dr. Weaver, I'd like to thank Dawn Hahn, who is Connie's trusted assistant, who supplied us with all the material we needed for her nomination. We kept this a secret from Connie, so she didn't know about the award until Dr. Bacchius walked into her office one day and told her. It gives me great pleasure to introduce distinguished Professor Connie Weaver, Department Head of Nutrition Science. Connie is the second female to be awarded this distinction. Connie received her BS and MS degrees from Oregon State University in the mid-1970s, and then earned her PhD at Florida State in 1978. She started her career at Purdue in 1978 and has been with us ever since. At the time of her nomination, Dr. Weaver's work was cited over 4,300 times, and her H factor, a measure of the depth and breadth of her impact, was 37. The basis of Dr. Weaver's nomination stems from her recent pioneering basic science on calcium metabolism. She has developed new techniques for using stable isotopes and kinetic models to study calcium distribution within living organisms. Her work serves as the foundation for our understanding of how diet influences bone health, especially in adolescence, but also at other ages. Specifically, she has implemented and validated the use of calcium-41 coupled with accelerated mass spectrometry as a means for long-term tracking of bone resorption in women. This collaboration with the Purdue Prime Lab permits one to measure minute amounts of calcium-41 and to follow the release of calcium from the bone into the blood, being able to capture the kinetics of bone resorption for the next 10 years within an individual. This work has been instrumental in setting new federal dietary guidelines for calcium. Dr. Weaver's basic science of calcium intake and metabolism also has resulted in her induction into the Institute of Medicine in 2010. Without further delay, it is a great pleasure to welcome the 2012 McCoy awardee, Dr. Connie Weaver, who will talk to us about chasing calcium into bones and beyond. Dr. Weaver?
Thank you so much for coming. I've really admired the work of other McCoy Award winners over the years and never really seriously imagined myself up here giving this lecture. So thanks to Howie and others who were involved in the nomination process. As you heard, the kind of work I do in bone is a problem, especially for women, because 80% of the hip fractures in postmenopausal women or individuals are in women. So that kind of led to launching this Women's Global Health Institute last March because of my interest in women's health. So that's our kind of logo for saying we're interested in preventing disease and promoting health throughout the whole lifespan. I want to tell you about some of the research you've heard about and introduce to you the main team who's been with me so long now. Dr. Bradine Martin runs the laboratory. Dr. Martin knows all the protocols for animals and humans and everything we do, and she's the actual one who keeps the organized group going and keeps the students on track with the protocols. And that's no easy task. I'm very grateful to her for that. We meet weekly with statistical geniuses who help us interpret the data, George and Linda McCabe, and that's the highlight of our week in lots of respects. Meryl Wasney, the kinetic modeler, Monroe Peacock, our very insightful clinician from IU School of Medicine, Anya, who runs the mineral analysis lab, Pam, who runs the animal studies, Doug Mesh, who runs the clinical protocols, and we've had various diet help over the years for a long time now. Jan Buckles and Olivia Wood before was the research dietitian, and Carl Wood, who runs the stable isotope analysis laboratory. NIH tells me it's really rare to have a clinical team intact for decades, and so we have accomplished quite a bit together, I think. Don Hahn, that you heard already provided Howie with materials, is the one who's developed this slide set, and you're going to see a little Don humor as we go through. So bones came late, after yeast and worms and flies. It wasn't until vertebrates, represented by the mouse, and humans that you actually had bones. Our group really approaches the study as from a whole body physiology perspective. And this is important, we think, because tissues have interdependence. Any tissue doesn't work in isolation, and although you have lots of genes in common with that mouse, 98%, the rest of the genes make a lot of difference, and the communication across tissues makes a difference. And I'm going to tell you about some of these tissue interactions that we've looked at. Also, whole body physiology is necessary to completely understand homeostatic regulation, like with calcium, and the whole body is necessary to understand the importance of timing, and I'm going to talk about that too. So homeostatic regulation. With bone, we're looking at remodeling, the changes that occur in the balance between bone formation through these specialized cells called osteoblasts and bone resorption through the specialized cells called osteoclasts. Now, osteoclasts are really unique in that they're the only cells designed to destroy the host tissue that they're in. And this remodeling is at enormous cost of energy. And so to introduce you to the processes that we study, I've got a Bone Biology 101 2-minute video.
Bone is a dynamic tissue that is continually being built, broken down, and rebuilt in a process called bone remodeling. Bone tissue is broken down and resorbed by multinucleated cells known as osteoclasts. These cells are derived from monocytes, which originate within bone marrow. Osteoclasts play an important role in liberating minerals and other molecules stored within the bone matrix. Bone tissue serves as a repository for vital minerals, including calcium phosphate and various biologically active molecules, such as growth factors. The release of calcium from the bone can play a role in maintaining its homeostasis within the body. The cells responsible for building new bone tissue are known as osteoblasts. Osteoblasts are thought to be derived from cells found to be associated with blood vessels. Once active, they start to produce the organic component of bone, osteoid, which is predominantly made of collagen. Minerals start to crystallize around the collagen scaffold to form hydroxyapatite, the major inorganic constituent of bone, which contains calcium phosphate. Bone mineral density, or BMD, can be used to estimate the strength of bone and to assess the risk of fracture. As osteoblasts form new bone tissue, many become embedded within the matrix and differentiate into osteocytes. The structure, composition, and cellular processes that occur within bone allow it to simultaneously serve as a calcium reservoir, while providing structural support for the vital organs and for locomotion.
So it's with calcium kinetics that we study this integrated process of remodeling from the activities of these cells. So this is a picture of the lifespan of bone mass. You have accelerated growth during adolescence, which is an important period I study. Then you have kind of a plateau when you have your adult peak bone mass, and then you lose bone with the loss of estrogen, especially at the time of menopause for women. So those areas of rapid bone turnover are when the remodeling process is not in balance. So during growth, you have bone formation exceeding bone resorption by a great deal, and in the period of bone loss, you have bone resorption exceeding bone formation. We really like to study those periods because we think you can have the most impact with interventions. So we modeled a bunch of data to see how long do you really have to deposit bone, to accumulate bone, forming your adult peak bone mass. And so in girls, we found out 91% of adult bone mass is already accomplished by age 17. And by age 19.8, you have 95% of your bone acquired. So if you think you can do much to acquire more bone after that, you're deluded. There's only a few percent of what you can do. So you're all over the hill in this audience about what you can do about building bone. So I want to talk about exercise as well as diet and what difference that makes in accumulating peak bone mass. So here's the team, and basically all my work is interdisciplinary teamwork, which is the most fun, I think. So you have Dorothy Teagarden, who is the project manager of this particular exercise intervention, Ben Hilberry in mechanical engineering, George McCabe in statistics, Con Johnston, who developed DEXA, the bone scans for humans down at IU School of Medicine, again, Monroe Peacock, and David Burr, a bone geometry expert. And what we learned is in these young college women in their 20s, we recruited them when they were sedentary, they're already losing bone. So that picture of a plateau to menopause isn't true if you're sedentary. We had to randomize to an exercise intervention to maintain the bone. So I'm thinking you really need to redraw this picture of the lifespan of bone mass. If you're sedentary, you're already going down. So once you hit menopause and you have that other hit, due to the absence of estrogen, you're well into the fracture risk zone by then. Well, let's bring calcium into the story. Calcium storage in bone is a functional reserve, and it's the only nutrient that can be said of. Every other reserve is for a rainy day for when you're not getting enough of that nutrient. But in bone, it has a mechanical weight loading function. Calcium is present as a constant percentage in bone mineral, around 31%. And because of that, following the fate of a calcium tracer reflects what's happening in bone, and we take advantage of that. Half of women over the age 50 will have a fracture, and the worldwide cost of fracture approaches $131 billion annually. there's a linear relationship between bone mass or bone mineral density and fracture risk. So maximizing peak bone mineral content during growth reduces the risk of fracture proportionally. So how we use tracers is we usually give an oral tracer to determine calcium absorption from the intestine, and an IV tracer then that follows all the fate, whether it's endogenous secretions back into the stools, through the kidney, into the urine, bone formation rates, bone resorption rates, what all is happening with calcium in the body. We start with absorption. So we've done a lot of tests from different sources of calcium to determine how well it's absorbed in different groups. We started with milk, or we may not have started there, but a long time ago we did labeled milk by getting a lactating dairy cow and putting 4 grams of heavy calcium tracer into the cow and collected the milk. And then we processed it and fed it to all different ages of women. And we learned that the teens, this is a current study we've got going now, this teen can absorb a glass of milk's worth of calcium at a rate of about 40%. The counselors who are in their 20s can absorb it at a rate of about 30%. But when you get as old as me, it's down around 25%. And we've studied the elderly, it can be as low as 5%. So your ability to absorb really dictates your needs. But on the other end of life, the efficiency of calcium absorption by a lactating from a lactating mother to the infant is up as high as 80%. So we labeled the lactating woman and measured this calcium absorption. We have studied all sorts of sources of calcium, from plant foods to animal sources to supplements, and we labeled the plants by growing them hydroponically and putting tracers into the hydroponic solution so they'd be taken up by the plants. And you can see there's a wide range in efficiency of calcium absorption, from low spinach up to high kale. But that's not the full story of what makes a good source of calcium. It's not just efficiency, it's the total content. So you would have to consume 4 or 5 cups of broccoli or kale to get the amount of calcium absorbed from 1 glass of milk. Probably not going to happen. Lately, we've been interested in the lower gut phenomena, because the upper gut is where calcium is usually absorbed from these foods. But we've found that some fibers that reach the lower gut can be fermented and produce short-chain fatty acids, lowering the pH, and then increasing mineral absorption. So if you won't drink enough milk or get enough calcium, there's hope for getting it to be more efficient. So we partnered with Cindy Nakatsu, who does the bacterial community profiles, and you can see the communities really segregate out by the amount of prebiotic fiber we give them. The hallmark probably of our lab is Camp Calcium, studying accumulation of bone and what factors influence that during the peak growing period of adolescence. So we determined that by running summer research camps and bringing adolescents in and renting a residence halls. And we've found, as you increase calcium intake, for every additional milligram of calcium absorbed, you prevent 1 milligram of calcium coming out of the bone. What a powerful effect diet has to prevent calcium loss from the bone. Here's data showing a range of calcium intakes, and skeletal calcium accretion keeps on increasing up to a plateau level, and then it levels off and you just excrete the rest. So where this plateau occurs is optimal calcium intakes for maximizing peak bone mass accretion, and that's 1,300 milligrams a day. And that led to my plate now saying 3 cups of milk is necessary to optimize bone health in adolescents. When they come in, they're consuming maybe 900 milligrams or less of calcium a day. We calculate if they got up to this optimal of 1,300 milligrams a day, over 1 year they could increase their bone mass by 4%, and over adolescence by 10%. Bongier calculates that kind of level of difference in peak bone mass could delay the onset of osteoporosis by 13 years and decrease the risk of fracture in postmenopausal women by 50%. We wondered, boys have bigger skeletons than girls, or men have bigger skeletons than women. Do they need more calcium? Here's the rate of skeletal growth in boys that occurs later than girls. They shoot up and get taller and accumulate more bones a little bit later, and the peak is higher to get that higher skeleton. So to ask the question, do you need more or are they more efficient, we brought boys to Camp Calcium for the first time in 2001 and matched them to the same sexual maturity as girls. And boys representing blue and girls representing pink, we found they were more efficient at accumulating skeletal calcium at every calcium intake by a constant amount of 171 milligrams a day. So the boys didn't need more calcium to get those larger skeletons. They're just more efficient at utilizing calcium. A similar story between black girls and white girls. At all levels of intake, blacks are more efficient at retaining calcium than whites. They accumulate more bone mass and have lower incidence of fracture. We modeled, the McCabe's modeled, how much do they need from onset of puberty of menarche to adulthood. And so if you compare the rate of calcium accretion during adolescence and project it on to adulthood, and you do the area under the curve, you would project that blacks would have 12% more bone mass as an adult than whites. And the national survey data show femoral neck bone mineral content and density are 10 and 13% higher in black women than white women. So it all happens in adolescence and predicted by our Camp Calcium studies. We've studied calcium requirements in all races, whites, blacks, and Asians, and just finished Mexican Americans. You can see by these pictures we have a lot of fun in camp, not just research. And the public impact, as you heard in the introduction, has been to influence calcium requirements in North America that are the same values for adolescents since 1997, used for the 2004 Surgeon General Report on Bone Health, and then used to help set 3 cups of milk or the equivalent for the dietary guidelines since 2005. One thing with the Clinical and Translational Sciences Institute and the hub for it that we've created with the help of Ann Christine Caitlin is now all the data are available to the public. So you can go to the Indiana CTSI Hub website, find the Camp Calcium website, and you can hit view and download all the data from all the camps since 1990 that we've had, all the individual data. And now investigators can download our data and do their own research projects with the data like a national resource. So let's move to fat and bone communication and connection. Katie Hill, when she was a graduate student here, went back and merged all the Camp Calcium data and asked the question, does BMI as a measure, body mass index as a measure of obesity, influence this relationship between calcium intake and skeletal calcium accretion? And it certainly did. As you increase obesity, you can, with enough calcium, acquire more and more bone. Not if you're down at the intakes most kids are, but if you got enough calcium into them, you would have proportionally more bone being formed, is what this data says, the higher you are in obesity. So we are thinking that really influences fracture. Before, we think fractures in kids are all about risk-taking. Here's my adventuresome son upside down in his snowboard there. No wonder he might get a fracture, right? But is it that, or is it relatively low bone mineral density? So they've known for a while that kids shoot up in height before they consolidate the bone mineral. And so there is a period of relatively low bone mineral density. So that's a peak risk of fracture in kids. But beyond that, the incidence of fracture in children has doubled in the last 3 decades. And also obesity has increased. So we think our data has explained that. they need more calcium to grow proportionally larger bone when they're obese, and this period of relatively low bone mass is exacerbated the more overweight they are. So in addition to having greater force when you fall, if you're overweight and you may be less physically active, low calcium intake probably really plays into low bone mass for the weight. Another predictor of calcium accumulation during growth and maintenance later in life is salt. Salt is not good for bones, and we studied black and white adolescent girls to try to see what is the quantitative relationship. And we think we've found that the handling of sodium and consequently calcium leads to the racial differences you see in osteoporosis and hypertension as adults. So we brought black and white girls to Camp Calcium that were matched for skeletal body size and sexual maturity, and we fed them either a low sodium diet first or a high sodium diet as salt, and then crossed them over, so they were their own comparisons. And we put the salt in Gatorade, drinks, or soup to get that kind of a large range of difference in salt. And we've learned a lot about racial differences and the effect of salt on first sodium retention and then calcium retention. So what happens at low sodium intakes, urinary sodium excretion is about the same between blacks and whites. But at the high sodium intakes, whites can excrete all that extra sodium by the extra salt, but blacks can't. So they go into a big net positive balance of sodium retention. And when you retain sodium, you retain water. So that could explain why blacks have a higher incidence of hypertension. But because sodium and calcium are linked transporters in the kidney for reabsorption, if you're excreting extra sodium, like the whites out on the high salt diet, it's dragging ions of calcium out with it. So high salt decreases your calcium retention or skeletal calcium accretion by quite a bit. So salt is leaching your calcium basically away from bones and out into the urine. But look at the racial difference. Blacks are clear up here on both sodium levels compared to whites. So even on the high salt diet, blacks are better off for utilizing the calcium than the whites were on the low sodium diet. So we think that explains why whites have an higher incidence of osteoporosis than blacks, and it's apparent early on. So what things have we learned about what regulates these periods of high turnover in the body? We have measured a lot of regulators, hormones, and different things that are thought to regulate bone accretion early, and by far the strongest one is insulin-like growth factor, IGF. That has as much explanation on the variance in or on calcium retention, about 25%, as did calcium intake at about 22%. We then tested an animal feeding study by giving IGF to see if we could manipulate how big the bones grew or how much calcium they accreted. And when they were in the rapid growing period, sort of like adolescents and kids, we could influence skeletal calcium accretion by 31% with IGF compared to the saline fed controls or administered controls. But just a little older, past their rapid growth period, there was no effect whatsoever of IGF in the older rats. Now, what about the other end? Or both ends. Why do sex steroid hormones control bones? Estrogen really influences bones here. IGF, I told you, influenced the onset of skeletal calcium accretion, but then what closes the epiphyses and stops the linear growth of bones is when the estrogen surge starts. So it stops the growth here, and then the absence of estrogen causes the osteoclasts to go wild and really increase bone resorption, so you have a loss. Recently, it's been shown that sex hormones work to control these bone changes through receptor-activated NF-kappa B ligand, or RANKL. It even has been shown to control when calcium releases into breast milk in lactating women. But we're going on to the postmenopausal women's stage of the life cycle now, and what our intention is to label bone with tracers of calcium, and you heard in the introduction, calcium 41 is what we use in people, and as people are going from normal healthy bone into osteoporotic bone as they're losing bone, if we pre-labeled the bone, we can follow the tracer out into the blood or the urine and measure quantitatively what the loss is and what might prevent the loss or accelerate the loss. So Emily Holman, my current graduate student, gave rankle to rats and found, relative to the controls, a really rapid increase in the amount of tracer that was coming out of the bone and into the blood. So that's not good for bones. Whereas the decoy receptor, OPG, had the opposite effect, immediately shut down resorption from the bone. So we're looking for strategies to intervene to have a high OPG to rankle ratio. If we can find dietary means to control that ratio, we can do a lot towards controlling bone loss. This method was developed by quite a team as part of the NIH-funded Botanical Research Center that we had here. The usual method for evaluating interventions to stop bone loss in postmenopausal women by the Food and Drug Administration, FDA, their approved intervention are 4-year randomized controlled trials looking at bone mineral density. So it's long, it's expensive, it takes hundreds of subjects or to thousands of subjects, and our method is based on the fact that bone turnover occurs at a rate of about 26% a year for the spongy trabecular bone and 3% a year for the cortical bone. So using this turnover rate and pre-labeling bone, we developed a 50-day screening method that's as powerful for showing efficacy of an intervention as the 4-year trial in 12 people in randomized order. So this is the accelerator mass spectrometer in the Prime Lab. George Jackson from physics measures calcium 41, this rare isotope, and it's so sensitive that once a person is labeled with a single small dose, we can follow their bone loss for the rest of their lives. So here's what a profile would look like in an individual. You give them the calcium 41 and you wait until the soft tissue calcium clears, until you have a steady state, and then you collect a few measures for baseline. Then you give an intervention, and if it goes down, that's effective in stopping the loss of calcium out into the urine. You've decreased it. Then you have a recovery period. Then you can test another intervention. This is a typical osteoporosis treatment drug. And you can compare then in the same subjects the relative effectiveness in just 50 days. So this shows you one publication we have where 0 is the person's own baseline, and estrogen, this is the net calcium retention of the tracer, stopped bone loss by 28%. And risedronate, another osteoporosis treatment drug, almost as good. Some dietary supplements had some ability to stop bone loss as well. They're not as effective as the drugs, but a whole lot fewer side effects. So far in the talk, I've been worrying about this end of the spectrum, where we're trying to see what can you give to avoid risk of inadequacy. How much calcium do you need? That sort of thing. Now we're spending some time on what is too much. There's a lot of concern about soft tissue calcification with all the calcium supplementation that's going on, and the data is not based on evidence, not strong mechanistic evidence. And we're trying to look at that now. And we're using a Purdue animal model that's here at Purdue, the Ossabaw pig model, which is the best model for metabolic syndrome and atherosclerosis, hypertension, all these things that humans get to frequently. And we're using calcium 41, so we have George Jackson measuring it in the Prime Lab. Alyssa Phillips is the graduate student on this. We have nutritionist and a pig nutritionist, Scott Ratliff from Animal Science, Sean Newcomer and Mike Sturrock are cardiac physiologists, Meryl Wasney is going to do the kinetic modeling, and Bill Van Alstyne the histology. And so we're expanding our model, not just to look at bone, but into coronary artery for soft tissue calcification and mammaries for breast cancer effects. So the remaining questions I want to spend part of my career answering is what controls calcium leaving the bone? How can we control that? And what is control of soft tissue calcification? I have a lot of people to thank. I've had wonderful postdocs in my laboratory. I've had fantastic PhD students. Master's students. Honors students. I'm not giving you the people that don't have a degree because we hire about 65 people for every Camp Calcium. And so we're a big employer on campus. Visiting faculty. I need to acknowledge the funding. Mostly it's been NIH. A few USDA grants. Industry, a lot of practical questions we've helped them answer. And I want to tell you about a legacy of a different kind that I want to leave at Purdue. My husband Lloyd and I have 3 wonderful sons, and Doug, the oldest, could be here, who's given us 7 wonderful grandchildren that we say are maybe future Boilermakers if we have anything to say about it. Thank you.
Questions for Dr. Weaver? Someone always has to start.
First, thank you, Connie, for such an enlightening presentation. The question I have is whether it matters we divide the amount of calcium we take over the 24 hours rather than take it in one shot.
Yeah, there's an inverse relationship between the load and efficiency. So kind of a rule of thumb is don't take more than 500 milligrams at a time because you've really decreased the amount you use, but it's really a linear relationship. Little bits all day long, not just all at once.
Just 2 quick questions. You probably said, but is calcium-41 a stable isotope?
So calcium-41 is technically a radioisotope, but the half-life's 100,000 years. And we give 50 nanocuries of it, so you can't measure it like a radioisotope. It behaves in lots of ways like a stable isotope. The dose load we give a person is equivalent to eating a banana for the amount of potassium-40 it would have in it. So it behaves much like a stable isotope.
And the other question is, like in a urine sample, if you're going to follow calcium, how many species? I mean, is it complex? Do you have to look at several different forms of calcium, or?
No, we ash it down and get rid of all the matrix, and so we don't worry about that. Precipitate it with oxalate, so you've got pure calcium oxalate in the end when you measure it.
Sorry, I don't have long enough arms.
Connie, how much does it cost to isotopically label a cow?
So this was done like in the early 80s, and 4 grams cost $30,000 for that amount. And so, yeah, we took real good care of that cow. There's someone.
Connie, in terms of the differences between the males and females in terms of bone absorption or calcium absorption in the bone, and even between the differences between whites and blacks, has there been follow-on to that to look at the enzymes, to look at the physiology further than the studies that you have done? Are there any genetic differences that we can start pointing to that say, oh, this makes you a better absorber or not?
So knowing the genetics in the area of bone has not been, once you have the genome mapped, has been very disappointing because no gene has explained more than about 1 to 3% of anything about bone mass. There is a gene that's been somewhat associated with calcium absorption. It's not believed by everyone because it's associational rather than linkage kind of information. So it's complex. It's not like a single gene's going to explain the race differences or sex differences. So race is a crude marker of genetics, but exactly what to go measure, yeah, it's still premature to know that.
So you had some nice data about how much the baby absorbed milk. My question was in relation to the mother and what you know about how much calcium she would absorb from what she takes in, because she is giving so much of that calcium into the breast milk.
That's correct. In the 3rd trimester of pregnancy and in lactation, you have accelerated, upregulated ability to absorb calcium. So interesting thing during lactation is women lose bone at a rate of about 1% a month, kind of, for a while. So you really take a dip in your bone mineral mass or density during lactation. And even calcium and vitamin D supplementation cannot influence that, stop it. But then when the baby is weaned, the mother upregulates her absorption again and comes back to pre-pregnancy levels. So we're programmed amazingly to recover from these things. So women who've had multiple children don't have lower bone density than women who've never had children.
In terms of bone quality, what's most important? Adequate vitamin D intake or adequate calcium intake?
Well, they're both important because vitamin D helps you utilize calcium. So what's most important depends on which one you're deficient in. So if you're deficient in both, they're both important to get because vitamin D helps you not need as much calcium. You can, with gobs of calcium, overcome vitamin D, because you'll learn in class next week that there's two paths for calcium absorption. One's not vitamin D dependent, and so you can just keep on taking more and not worry about the vitamin D for bone. But vitamin D is finding uses in other tissues, so just because you have enough for calcium absorption doesn't necessarily mean you have enough to protect you against cancer or multiple sclerosis or hypertension or other things.
Connie, I wonder if you would speak just very briefly about estrogen replacement in postmenopause women. And, we all know that it would be much better if we could accomplish this through dietary means. But, the trend maybe 10, 15 years ago was to recommend estrogen replacement. And I just wonder if you'd speak to that just very briefly. I know it's a complicated issue.
So this slide sort of summarizes what I'd want to say about that. Estrogen really works. So you have the normal rate at baseline of bone loss or calcium retention, and then estrogen really stopped the calcium loss from the bone and protected its loss. And the Women's Health Initiative also showed it really reduced hip fracture incidence. But because there was a slightly elevated risk of breast cancer, all of the societies stopped recommending estrogen long term. I mean, it's the only really good thing for stopping hot flashes. So physicians will prescribe estrogen to women up to a year because of the hot flashes, but not long-term use for bones because of this slightly elevated, like 6 to 8 out of 10,000 kind of increase in breast cancer and some other side effects. So they say go to the bisphosphonates. That's been the strategy for bone, which also works about as well as estrogen. But now you have atypical femoral fractures and osteocrosis of the jaw, and so people are becoming worried about bisphosphonates and starting drug holidays and things to try to deal with that. And of course, that's completely untested, whether that helps you or not. But it's a strategy to try to help with side effects. So yeah, we would like some simple dietary things, and probably we're going to find they're not as impactful, whatever we study, maybe not as impactful as the estrogen or the drugs, but they're not going to have the same side effects. Although in an animal study, we found amazing comparisons to estrogen and bisphosphonates, I should say, with plum powder and blueberry powder. So we're hoping to study that further. We haven't studied it in humans yet. But maybe there is something dietary out there that's good for you in the berry class. Maybe it's time to go party.
Well, please put... Please join us across the hall in the gallery for reception.